We're speaking this morning with Dr. Gregory Hannon of Cold Spring Harbor Laboratory, who is also an investigator of the Howard Hughes Medical Institute. Greg and members of his team here at Cold Spring Harbor have been involved in a very interesting project, which, among other things, has led to a complete genome sequence, which is being published today, for Neanderthal. We wanted to ask Greg about his team's involvement and what they contributed to this larger effort. Greg, if you could tell us first, if you don't mind, what's the sort of headline news in the part of the project that you were involved in?
Well, so it was one set of Neanderthals that if we gave them a shave and a haircut, put them in a suit, stuck them on the New York subway, that no one would notice them. And what we found is that that is also true of their genomic sequences, that if you gave their genomic sequences a shave and a haircut, that it would be almost indistinguishable from the genomic sequences of modern humans, at least at the level of the proteins that are encoded by the genome.
Mm-hmm. And I imagine this was pretty surprising to you, or am I wrong about that?
No, it was quite surprising, and I think, in part it speaks to our deep-rooted biases, that there's something unique, something very special about modern humans, that these capabilities that we don't share with any other animal on the planet make us special, and that specialness that we ascribe to our species must also be reflected at the level of our genome.
Mm-hmm. How did it happen that you became involved in this work? A lot of the work that you're very well known for in the scientific community has to do with RNA. How did it happen that you got involved in a project about the Neanderthal genome?
Well, it was an interesting occurrence and an interesting comment about how modern science works. It turned out that we had developed a technology for completely different purposes, in part for studying the genomes of cancer cells, that could solve one of the major problems, one of the major barriers in the study of ancient DNA, which is that DNA is often contaminated very heavily with DNA derived from the environment. These are samples, bone fragments that have lain in caves for 40,000 years, and over that time they've been infested with bacteria and fungi, etc. And that environmental contaminant has overtaken very often the DNA of the animal, the individual, from which the bone was derived.
So we will speak separately with a member of your team, Emily Hodges, Dr. Emily Hodges, who's a postdoc in your lab, who has worked on this technique.
Well, she really developed that.
She did indeed. I didn't mean to slight her. She developed this technique, and you have used it for other purposes over the years. How did it apply in this case? What did it enable you to do relative to that contaminated Neanderthal sample that you obtained from a team in Germany?
Well, Svante Pääbo actually called us initially about another project, which was sort of much more conventional. It was the study of a modern genome.
And Svante Pääbo, tell us who he is.
Svante Pääbo is probably the world's foremost expert on ancient DNA. He's at the Max Planck Institute for Evolutionary Anthropology in Leipzig, and he is the leader of this international effort to sequence the Neanderthal genome, an effort that's been going on, I think, since the 90s.
And he contacted you in regard to this contamination problem?
No, he contacted us with regard to the study of a modern genome, trying to identify a particular mutation in a strain of rats that they'd been studying, a mutation that was linked to a particular behavioral phenotype. And really it was that initial contact that led to our involvement in the broader Neanderthal genome project.
So once you got this sample from Dr. Pääbo, what is it that you were able to do with it that his team was not able to do with it?
Well, so these ancient bones contain a very small percentage of hominid DNA, somewhere around 1% in the best of all possible cases. And it's those samples, those bone fragments that contain 1% of hominid DNA, which are, sort of that's the gold. This is what they're using to do the draft sequence of the entire genome. Now, what that means is that for every stretch of hominid DNA that you sequence 100 stretches of bacterial or fungal DNA. Now, obviously, with samples that are 99% contaminant, it costs 100 times as much to sequence the genome as it does if you have a pure sample. And what we felt our technology could do, although we had to prove it, was to enrich the hominid fraction of DNA within that contaminated sample to a very high percentage, 60, 70, 80% of the total sample, which enabled both a reduction in cost and a much higher accuracy in the sequencing. Now, initially we did this using material from one of the bones that was 99% contaminated, but the paper is actually published on a sample from another Neanderthal, one that lived about 43,000 years ago in Spain, which was considered unsalvageable for molecular biology because the contamination levels there were not 99%, but something like 99.9%. So one fragment of hominid DNA per 1,000 fragments of total DNA.
And the technique that Emily, in concert with others in your lab, developed has a way of isolating certain segments within this 1 out of 1,000 fraction.
Correct, and so she was able to enrich particular pieces of hominid DNA almost 200,000 times from the original sample.
That's remarkable. And so once you were able to get the sample and apply this technique, what did you find? It's quite remarkable what you did find, as you said earlier. Take us through it a little bit.
Well, what we were trying to do was to understand the evolutionary relationship between the proteins that are encoded by our genome, the proteins that are encoded by the Neanderthal genome, and the proteins that are encoded by our closest living relative, the chimpanzee.
And just explain for a second why the proteins. Why are you attacking it from the protein level?
Well, proteins are the functional elements of the cell.
Mm-hmm.
And these are the product of the genome that does work, and variations in protein capabilities can create variations in structure of organisms, in the capabilities of those organisms, differences in how neurons develop, differences that are really underlying the unique capabilities of modern humans. And the hope was that this might speak to some of the most intriguing issues of our own lineage. If you think about the differences between modern humans and Neanderthals, what we're curious about is, how did they think? Could they speak? Did they have culture? And so the notion was that perhaps by understanding specific differences in our protein coding genes, we might identify changes that we could attribute to some of our unique capabilities. To give one example, the Pabo group had found a number of years ago that a particular gene called FOXP2, that a particular mutation in those genes that was linked to language was found both in modern humans and in Neanderthals. Now that doesn't mean that Neanderthals spoke, but it means that they had this particular gene variant that is uniquely correlated with speech.
And so in doing this comparison where you're, pardon me, situating Neanderthal on the continuum between chimp at one far end 6.5 million years ago and modern human today, again, to return to the finding, you found that the Neanderthal protein, the set of proteins, the proteome of the Neanderthal, is almost identical to the modern human proteome.
That's correct. If you consider chimps and modern humans, our most recent common ancestor was 6.5 million years ago. 3/4 of all the proteins encoded by the chimp genome and the human genome have at least one difference. If we compared a single Neanderthal to a single human individual, the so-called human reference genome that emerged from the Genome Project, there were about 1,000 proteins that were different. But then, if we compared the Neanderthal individual, just one individual, to the diversity of human populations, that initial number of 1,000 differences to one reference human dropped to only 88 differences in 83 proteins.
So where do you go from this finding, this very remarkable finding about the similarity of Neanderthal and modern human?
Well, where we go is probably not a lot farther. We're itinerants in this field. We came into this collaboration, which was really just a wonderful privilege, and contributed what we could contribute. But the field as a whole now has to understand whether changes in these protein coding genes lead to phenotypic differences, lead to the differences between humans and Neanderthals, or whether it's something else in the genome that does this.
Thanks very much, Greg, for talking to us today.